Introduction. One of the most important concepts in biology is that the basic unit of structure and function in living organism is the cell. It is the smallest unit that can carry out all activities of life. Cells are the building blocks of complex multicellular organisms. The modern theory of cellular organization states that all living organisms are composed of one or many cells. 2. All new cells arise from division of pre-existing cells. 3. Cells contain the hereditary material of an organism which is passed from parent to daughter cells. Number 4. All metabolic processes take place within cell. Techniques used in cell biology. Microscopy makes a very valuable contribution to our understanding of cells. However, techniques are also needed if the function of organelles are to be studied. Various techniques have been used for isolating and examining various cell components of these different staining, centrifugation, tissue culture, chromatography, electrophoresis, etc. are the most common techniques. The examination of a cell and its component depend upon the magnification power of two convex lenses to produce magnified image of a very small object. Tissue culture. Tissue culture is the growth of tissues and cells separate from the organism. This is typically facilitated by the use of liquid semi-solid growth mediums such as broth or agar. Tissue culture produces clones in which all the product cells have the same genotypes unless affected by mutation during culture. In the following section, a list of apparatus and main steps of tissue culture are given. Tissue culture apparatus. Cell culture, hood, incubator, water bath, centrifuge, refrigerator and freezer, cell counter, inverted microscope, liquid nitrogen, sterilizer. Main steps in tissue culture. Selection of the plant tissue from a healthy, vigorous mother plant. This is often the apical bud, but can be other tissue. This must be sterilized to remove microbial contaminants. Establishment of the explant and a culture medium. Medium sustains the plant cells and encourages division. It can be solid or liquid. Each plant species and sometimes the variety within a species has particular medium requirement that must be established by trial and error. Multiplication of the egg plant gives rise to a callus, a mass of loosely arranged cells which is manipulated by varying sugar concentrations and auxin, low cytokinin, high ratios to form multiple shoots. The callus may be subdivided a number of times. The shoots are transferred to a growth medium with relatively higher auxins cytokinin ratios, which results in the formation of roots. Plantlets are deflosked and hardened up by gradually decreasing the humidity. This is necessary as many young tissue culture plants have no waxy cuticle to prevent water loss. Electrophoresis. It is a technique used to separate molecules of different electrical charges under The influence of different electrical fields, negatively and positively charged ions molecules will move towards anode and cathode respectively. Two factors affect the speed with which charged molecules move toward an electrode. The amount of charge, greater charge, faster movement, and vice versa. The size of molecules, smaller molecules, faster movement, and vice versa. Cell wall and plasma membrane. The cell wall was discovered by Robert Hooke in 1665 earlier. Then the protoplast. It is the outermost boundary of the plant cell. It is secreted by protoplasm of the cell. Its thickness varies in different cells of the plant. Each cell, whether isolated or occurring in tissues, has its own cell wall. Cell wall of neighboring cells are held together by an intercellular substance called middle lamella. Cell wall varies greatly in morphology and chemical composition, but its characteristics have a close relation with age and function of the cell on the basis of development and structure. The cell walls have three fundamental parts, namely middle lamella, primary wall, secondary wall. Middle lamella cements together the wall of neighboring cells. It is usually thin and about 1 micrometer in thickness. In woody tissues, the middle lamella is commonly lignified. Primary wall is the first wall formed in a developing cell. It is usually 1 to 3 micrometer in thickness. More or less elastic and extendable, crystalline and optically active. It is composed of cellulose, pectic compound, mostly polysaccharides, and hemicellulose. The secondary wall follows the order of primary wall in development. It is laid down inside the primary wall. Its thickness is about 5 to 10 micrometer more or less rigid, crystalline, and strongly optically active. It is mainly composed of lignin, cellulose, non-cellulosic, polysaccharides, hemicelluloses, and mineral salts of calcium, magnesium, potassium, and some silica. A major rule of the cell wall is to form a framework for the cell to prevent overexpansion. Cellulose fibers, structural proteins, and other polysaccharides helps to maintain the shape and form of the cell. Plasma membrane. Plasma membrane or cell membrane as the outermost boundary of the animal cell and inner to cell wall in plant cells. Cell membrane as chemical of lipids 20 to 40% and protein 60 to 80%. In addition, there is a small quantity of carbohydrates present. Many biologists contributed to establish the structural organization. The modern technology has revealed that lipid bilayer is not sandwiched between two protein layers. In 1972, Singer and Nicholson proposed a most acceptable model for membrane called fluid mosaic model. This model is in agreement with photograph of cell membrane by electron microscope. This model explains that membrane is like a sea of lipids in which proteins are floating. Rule of protein. The protein are not arranged in sheet but as globes of protein which are floating about in the sea of lipid molecules. Some proteins extend completely through the double layer of lipids from one side to the other side and are called intrinsic proteins. Some other proteins are smaller and are placed among the phospholipids molecules. These are on one side of membrane and are called extrinsic proteins. Carbohydrates extend out from the outer surface of the membrane and are attached either to membrane lipids, glycolipids, or proteins as glycoproteins. Rule of glycolipids and glycoproteins. Lipids, proteins, and carbohydrates are responsible for functional diversity of the membrane. Lipid bilayer makes the membrane differentially permeable barrier that allows the transport of non-polar material across it and prevents ionic materials. Membrane proteins, on the other hand, makes a selectively permeable barrier that select materials according to cell need. If glucose concentration inside the cell is proper, no more glucose can enter the cell. Extrinsic protein function as receptor that receives the stimuli from the environment and thus inform the cell to respond. Integral protein, also called as permeases, regulate diffusion, osmosis, and active transport of ionic materials. Membrane carbohydrates such as glycolipids and glycoprotein provide receptor sites that receive different types of stimuli like hormone receptor sites, receives nerve impulses, recognition of antigen and food materials, thus inform cell for response. Membrane carbohydrates are also responsible for endocytosis, phagocytosis, and pinocytosis. Thus, glycolipids and glycoproteins act as cell surface makers. Rule of plasma membrane. Plasma membrane is a dynamic structure only about 7 nanometer wide, but it is present barrier to the movement of ions and molecules, particularly polar molecules such as glucose and amino acids, which are repelled by the non-polar hydrophilic lipids. This prevent the aqueous content of the cell from escaping. However, transport or maintenance across membrane must still occur for a number of reasons. For example, to obtain nutrients, to excrete waste substances like urea, uric acid, etc., to secrete useful substances, hormones, enzymes, etc., to generate the ionic gradients essential for nervous and muscular activity, to maintain a suitable pH and ionic concentration with the cell for enzyme activity. Cytoplasm. The living contents of eukaryotic cells are divided into nucleus and cytoplasm, the two together forming the protoplasm. Cytoplasm is aqueous water-containing substance containing a variety of cell organelles and other structures such as insoluble waste and storage products. The soluble part of cytoplasm forms the ground substance between the cell organelles and is called cytosol. It is formed of about 90% water and forms a solution which contains all the fundamental biochemical of life. In cytosol, small molecules and ions may form true solutions, and some large molecules form colloidal solutions. A colloidal solution may be a solid, non-viscous, or a gel, jelly-like, or viscous. Often, the outer region of cytoplasm is more gel-like. Apart from acting as storage of vital chemicals, the ground substance cytosol at the site of certain metabolic pathways such as glycolysis and living cells. The cytoplasm contains several cell organelles such as endoplasmic reticulum, mitochondria, Golgi complex, nucleus, plastids, ribosomes, lysosomes, centrioles, and various others. In the cytosol, mitochondria move about in cytoplasm due to cytoplasmic streaming movements. This is an active mass movement of cytoplasm, which is called cyclosis. This is typical plant cell. Cytoplasmic organelles, endoplasmic reticulum, most important topic. Under the electron microscope, a network of a channel is seen running through cytoplasm of all eukaryotic cells. These channels are often continuous with plasma membrane and also appear to be a contact with the nuclear membrane. The whole system of channels is said to be endoplasmic reticulum. These membranes vary widely in appearance from cell to cell. These channels are separated from the cytoplasmic materials by the spherical or tubular membrane, one above the other called cisternae. There are two morphological forms of endoplasmic reticulum: a rough form with attached ribosomes and a smooth form without ribosome. Endoplasmic reticulum are mostly present outside nuclear membrane and thus involved in protein synthesis. After synthesis, the proteins are either stored in the cytoplasm or exported out of the cell through these channels. The smooth endoplasmic reticulum helps in metabolism of a number of different types of molecules, particularly lipid synthesis. They also help to detoxify the harmful drugs. In some cells, smooth endoplasmic reticulum is responsible for transmission of impulses. For example, muscle cells, nerve cells. In addition, smooth endoplasmic reticulum play an important role in the transport of materials from one part of cell to the other part. It provides mechanical support to cell so that its shape is maintained. Ribosomes. These are tiny cells organelles about 20 nanometer in diameter and were first studied by Hlad 1955. Eukaryotic ribosomes are composed of almost equal amount of RNA, ribonucleic acid, and protein. Hence, they are ribonucleoproteins particles. The RNA present in ribosome is ribosomal RNA. Ribosomes exist in two forms, either freely dispersed in the cytosol are attached with rough endoplasmic reticulum as tiny granules and are the site of protein synthesis. Ribosomes are sized in nucleus of the nu- nucleolus of the nucleus. An example of protein synthesized by free ribosome as hemoglobin in young red blood corpuscles. Each ribosome consists of two subunits, one large and one small, as shown in figure. Sedimentation has revealed two basic types of ribosomes called 70S, 50 plus 30S, and 80S, 60 and 40S ribosomes used in ultracentrifugation. 70S ribosomes are found in prokaryotes, while slightly larger 80S in eukaryotes. The two subunits on attachment with each other require magnesium ions. The ribosomes are to messenger RNA through small ribosomal units. A group of ribosomes attached to messenger RNA called polysomes. Golgi apparatus. The Golgi apparatus was discovered by Camillo Golgi in 1898 using special staining techniques. This apparatus, which was found in all eukaryotes, consists of stacks of flattened membrane bounded sacs called cisternae. These cisternae, together with associated vesicles, are called Golgi complex of interconnected tubules around the central stack. The Golgi complex consists of units called dictysomes. Each dictysome is formed bundles of curved and flattened cisternae, associated tubules, and secretory vesicles. Dictysomes have two distinct faces. The proximal or forming face present close to nucleus, and a distal trans or maturing face located towards the cell membrane. Vesicles and tubules fed up from rough endoplasmic reticulum flow, converge, and fuse with forming face to form new cisterna. Function of Golgi apparatus. Golgi apparatus helps in the storage of secretory products and in modification and packaging of secretory products. In some cases, polysaccharides may be synthesized from simple sugars in the Golgi apparatus. These polysaccharides may then be attached to proteins and lipids to form glycoproteins and glycolipids. Secretory vesicles produced by Golgi apparatus may play an important role in adding surface area to the plasma membrane. Lysosomes. Lysosomes are cytoplasmic organelles found in most eukaryotes and are different from other cytoplasmic organelles. Due to their morphology, these were isolated as separate components for the first time by Judd in 1949. They are surrounded by a single membrane and are simple sacs that contain very large variety of food digesting enzymes called hydrolysis. Any foreign object that gain entry within the cell are immediately engulfed by lysosomes and are completely broken down into digestible feces. This process is known as phagocytosis. They are very abundant in those animal cells which exhibit activity, for example, neutrophils. Lysosomes are bounded by a single membrane and contain numerous hydrolytic and acid phosphate enzymes. These enzymes are synthesized and run endoplasmic reticulum and are further processed in Golgi apparatus. The processed enzymes are bodied up as Golgi vesicles and are called primary lysosomes. These contain those enzymes which can digest the phagocytosed food particles during autophagy. Some of old, worn-out parts of the cell are also digested, and this way, materials of cell may be recycled and cell may be renewed. Their enzymes can also result in degeneration of cell, as many as may occur during the development processes. Malfunctioning of lysosomes. Several congenital diseases have been found to be due to accumulation of substances such as glycogen or various lipids within the cell. These are also called Tourette diseases and are produced by mutation that affects one of the lysosomes in the metabolism of certain substances. Peroxisomes Peroxisomes are small subcellular bodies of approximately 0.1 micrometer in diameter surrounded by membrane and found in a great variety of organisms, including plants and animals, which are also called microbodies. In animals, they are most common in liver and kidney cells. They are similar to lysosomes, but they are somewhat more dense and have different enzyme systems. For example, the enzymes of peroxisomes are known to break down hydrogen peroxide into oxygen and water, protecting cells from its corrosive effects. In the leaves of green plants, photorespiration may occur in peroxisomes. Glycosomes are present in flowered cells. It contains a number of enzymes including glycolic acid, oxidase, and catalase. These organelles are most abundant in plant seedlings. The primary activity of glycosomes is the conversion of fatty acids to carbohydrates and lipids. Such seeds, such as soybeans, lysosomes provide sites for the breakdown of lipids. These organelles are absent in seed for in lipids. Cytoskeleton. Kolzap in 1928 by his microscopic studies suggested the existence of an organized fibrous network of skeleton in cytoplasm of eukaryotic cell. Later on, Cohen 1977 confirmed the waves of Kolzap by his electron microscopic studies. According to Cohen, the cytoplasm of eukaryotic cells contain a cytoskeletal network of different types of microtubule, microfilaments, and intermediate filaments. Microtubules. These are long, unbranched, slender, tubulin protein structures. One very important function of microtubule is their role in assembly and disassembly of the spindle structures during cell division. Microfilaments. These are considerably more thin cylinders made of contractile actin proteins linked to inner face of plasma membrane. They are involved in cyclosis. Intermediate filaments. They have diameter in between those of microtubules and microfilaments help in maintaining the cell shape. Centrioles. Animal cells of some microscopic organisms and lower plants, for example, mosses, liverworts, contain two centrioles located near the surface of the nucleus. These are small hollow cylinders about zero point 0.3 to zero point 0.5 micrometer long, about zero point 0.2 micrometer diameter. They occur in pairs. In cross-section, each centriole consists of cylindrical array of 9 microtubules. However, each of the 9 microtubules is further composed of 3 tubules. The two centrioles are usually placed at right angle to each other just before cell division. A centrioles duplicate and one pair migrate to the opposite side of the nucleus. Spindle of the nucleus. Spindle fibers formation takes place by using centrioles located inside nucleus. They are absent in higher plants. Centrioles play an important role in the location, forming during cell division, and in formation of cilia and flagella. Mitochondria, most important topic for examination. Mitochondria are found in the cytoplasm of all eukaryotic cells. Although in highly specialized cells such as mature RBCs they may be absent, they were first seen in granules in muscle cells. In 1850, they are known as the powerhouse of the cell. Under electron microscope, mitochondria appear to be vesicles, rods of filaments, and also show complex morphology. Although their number, shape, and internal structure vary widely, mitochondria is bounded by two membranes. The outer one is smooth, while the inner membrane forms enfolding into inner chamber called mitochondrial matrix. These enfold are called cristae. The membranes have the same chemical nature as that of cell membrane. The presence of ribosomes and DNA inside mitochondria indicate that some proteins are synthesized in it, so it is self-replicating organelle. The inner surface of cristae in the mitochondrial matrix has small nub-like structure known as elementary particles. As mitochondrion as aside for all the reaction of aerobic respiration, its matrix contain a large number of oxidative enzymes, coenzymes, organic and inorganic salts vital for aerobic respiration, Krebs cycle, fatty acid metabolism. As a result of these metabolic processes, the energy extracted from the organic food is transformed into energy-rich compounds ATP, adenosine triphosphate, and in turn, this energy is provided to the cell on demand. Plastids. Membrane-bounded, mostly pigment-containing bodies present in the cell are called plastids. Plastids are unique organelles found in cells. There are three main types of plastids: chloroplasts, chromoplasts, leucoplasts. Chloroplast. In prosynthetic plant cells, these are membrane-bounded structures containing a green pigment, chlorophyll. Chlorophyll is organic compound which helps the cell to absorb sunlight and utilize it into manufacture food. Chloroplasts vary in their shape and size, with a diameter of about 4 to 6 micrometer. Under electron microscope, they appear to be heterogeneous structures with small granules known as grana embedded in matrix. Chloroplast shows three main components: the envelope, stroma, and the thylakoid. The envelope is formed by a double membrane, while stroma covers most of volume of the chloroplast. Stroma is fluid which surround the thylakoids. It contains protein, some ribosomes, cellular DNA, Eh, It is in this part of the chloroplast where carbon dioxide is fixed to manufacture sugars. Thylakoids are the latent vesicles which arrange themselves to form grana. A granum appears to be a pile of thylakoids stacked on each other like coins. On average, there are 50 or more thylakoids piled to form one granum. On the layers of thylakoids, chlorophyll molecules are arranged, and that is why granum appears to be green. Each granum is interconnected with one green part called intergranum. Chloroplasts are also self-replicating organelles like mitochondria. Chromoplast. Chromoplasts impart color to the plant parts other than green. They are present in the petals of the flower and in the repent roots. They help in pollination and dispersal of seeds. Leucoplasts. Leucoplasts are colorless. They are triangular, tubular, or of some other shape. They are mostly found in the underground parts of the plant and store food. Nucleus molecules was reported in 1838 by Robert Brown. Its early discovery was due to its prominence in many cells, where it stands out as slightly darker than the surrounding cytoplasm. They are typically about 10 micrometer in diameter. It is one of the most important organelles because it controls all the metabolic activities and has the genetic information in the form of chromosomes and DNA. They may be irregular or spherical in shape. Generally, the cells having one molecule are mononucleate. Those with two nuclei are binucleate, and with more than two nuclei are said to be multinucleate. Parts of nucleus. Nucleus consists of the following parts: nuclear membrane, nucleolus, nucleoplasm, chromosomes. Nuclear membrane. The outer membrane of the nucleus is the nuclear membrane which separates the nucleus from the cytoplasm. The nuclear membrane is actually the nuclear envelope composed of two membranes. The outer membrane is continuous with the endoplasmic reticulum, while the inner membrane encloses the nuclear contents. These membranes are not continuous, having subtle pores at points. The nuclear pores allow the exchange of materials between the nucleus and the cytoplasm. Nucleolus. It is darkly stained body within the nucleus and is without any membranous boundary to separate it from the rest of nuclear material. There may be one or more nuclei in the nucleus. The ribosomal RNA in the nucleolus it is composed of two regions. Peripheral granular area composed of precursor of ribosomal subunits, and a central fibrillar area consisting of large molecular weight RNA and rDNA. Nucleoplasm. The nucleoplasm is a colloidal mixture of organic and inorganic salts and ions. It serves the matrix in which nucleoprotein complex chromatin is suspended. It also serves as storage place for enzymes, raw material needed for DNA replication and synthesis of RNA. Chromosomes. During cell division, chromatin material stand as dark thread-like structures known as chromosomes. Under the compound microscope, chromosomes appear to be made of arms and centromeres. Centromere is the place on the chromosome where spindle fibers are attached during cell division. Each chromosome consists of two identical chromatids held together at the point kinetochore, a centromere. A chromosome is composed of DNA and proteins. All the information necessary to control the cell activities is located on the chromosomes in the form of genes which are transferred from one generation to another. The number of chromosomes and all individuals of the same species remains constant generation after generation. Diploid and haploid number of chromosomes. The pool number of chromosomes in normal body cells is diploid 2n, whereas haploid n is the half number of chromosomes present in germ or gamete cells. For example, human sperm and eggs have 23 each, and those of Drosophila have 4 each sperm and eggs. So, in this way, after fertilization, the number of chromosomes remain constant in the next generation. Prokaryotic and eukaryotic cells. Biologists have divided cells into two types, prokaryotic and eukaryotic cells. The differences between these two types of cells are mainly based upon the structure of their nuclei. Eukaryotes have a very well defined nucleus in which nuclear material is enclosed in double nuclear membrane. In prokaryotic cells, the genetic material in the chromatin network is without any nuclear membrane and is directly submerged in the cytoplasm. Prokaryotes include bacteria and blue-green algae, while eukaryotes include all other unicellular or multicellular organisms such as protists, animals, plants, and fungi. Test studies show that prokaryotic cells generally lack many of the membrane-bounded structures which are present in eukaryotic cells. For example, mitochondria, endoplasmic reticulum, chloroplast, Golgi complex are absent in prokaryotes. Since there is no nuclear membrane and prokaryotic cell has no distinct nucleus and its DNA molecule is directly suspended in the cytoplasm. Prokaryotes have small-sized ribosomes. 70S in prokaryotes mitosis is missing and the cells divide by binary fission. Because of simpler structure of prokaryotes, it was widely accepted for a long time that prokaryotic cell represents a more primitive stage of evolution than eukaryotes. Perhaps the most distinctive features of the prokaryotic cell is its cell wall composed of polysaccharides chain bound covalently, a shorter chain of amino acid forming peptido or murein. The cell wall is often regarded as a single huge molecule or molecular complex called murein. The cell wall of plants, eukaryotes, is generally made of cellulose and is differently structured than that of bacterium prokaryotes. Structure of bacteria as model prokaryote. Bacteria, as mentioned earlier, are prokaryotes. These are smallest cellular organisms and are most abundant in the universe. Bacteria, along with cyanobacteria, which are included kingdom prokaryote, are the only living prokaryotic organisms. The cell wall of bacteria has membrane. It is rigid structure and determines the shape of the bacterium. It also protects the cell osmotic lysis. Bacterial cell, unlike eukaryotic organism, lacks discrete chromosomes and nuclear membrane. The nuclear material DNA in bacterial cells occupies a position near to center of the cell. This material is a single, circular, and double-stranded DNA molecule. Bacteria only have ribosomes composed of RNA and proteins. There are thousands of ribosomes in each healthy growing cell. Sexual reproduction in bacteria involves exchange of DNA. This process occurs in three different ways. Conjugation surface upon bacterium and travels to another bacterium. Bacteria essentially exchange DNA via conjugation. Transformation. Transformation bacteria pick up pieces of DNA from their environment. Transduction. In transduction, viruses that infect bacteria carry DNA from one bacterium to another. Key points. Cell as the unit of structure and function of living organisms. The process by which various cell components can be isolated is called Cell fractionation. The cell theory describes all organisms are composed of one or more cells. The cell arises from pre-existing cells, and the cell is the structural and functional unit of all organisms. The cell may be prokaryotic or eukaryotic. The prokaryotic cell lacks most of the membrane-bounded organelles. The nuclear region in prokaryotic cell is called nucleate. All cells are bounded by the plasma membrane. Cell wall is an additional covering and plant cells outside the plasma membrane. The model of plasma membrane which is accepted the world over is fluid mosaic model. Plasma membrane, a selectively permeable membrane. Cell wall in plant gives shape, protection and support to the cell. Endoplasmic reticulum helps in the transport of material between the cytoplasm and nucleus and also involved in the protein synthesis. Golgi apparatus is mainly concerned with storage of secretory products and are made up of vesicles arranged approximately parallel to each other. Lysosomes are bounded by a single membrane. Mitochondria are the energy producing organelles, hence called powerhouses of the cell. Plastids are of three types: chloroplast, chromoplast, and leucoplast. Microfilaments and microtubules give support, shape, and strength to the cell. Most cells contain a pair of centrioles lying at right angle to one another. The largest and the most conspicuous structure are cell organelles as the double membrane and bonded nucleus. The two prominent structures present in the nucleus are chromosomes and nucleolus, nucleoli.